All right, thanks everyone. Thanks to ARM for giving us the opportunity to talk about Portal and what we're doing over there. My name's Anil Narasimha. I'm the Chief Commercial Officer. Our goal is to shove things into cells, ex vivo. And so our two primary application focus areas are drug discovery and cell engineering, cell therapy. So obviously I'll talk about the cell therapy aspect today. But the idea is kind of being agnostic to the type of cargo that's being delivered into the cell and the type of cell itself. And so what is our strategy? Basically, it's super simple. It's you take your cells of interest, your cargo of interest, and then you pass it through a thin silicon filter or membrane that has a bunch of pores in it. And the pores are about 70 to 80% of the cell size. And so as the cells get flown through the system, they get squeezed, and that squeezing causes a temporary permeability in the cell membrane. And so during that 30 seconds, any type of cargo can be uptaken by simple diffusion. We're a startup. We're about 2 years old now, but we already have 50 partners placing our devices there, including a lot of the large pharma. I'm going to talk about a couple of the products that we have. One is the research scale, what we call the gateway system. We also have a high throughput system that we launched earlier this year at SLAS, where we were fortunate to win the new product award. And then our clinical scale integration that I'll talk about as well. Just to kind of give you a comparison of kind of the other approaches that many people are familiar with, whether it's viral vector approach, LNPs, electroporation. What our approach is unique about is it's first integratable with other systems, meaning you can integrate this into liquid handlers or cell processing units that I'll talk about in just a second. And then obviously kind of the other metrics that a lot of people use, whether it's efficiency, viability, can you multiplex delivery, meaning can you deliver multiple cargoes at the same time? Is it agnostic or is it universally efficient and viable for a variety of different cell types and cargoes. So we wrote a paper about kind of looking at the landscape of transfection technologies, and typically what we kind of, what's kind of out there right now is if you're doing DNA or let's say nucleic acids into cell lines, that's pretty easy to do. It becomes a little more finicky when you're talking primary cells, patient-derived cells, and then there's that third column in red where it's non-nucleic acids. So we're talking, proteins, peptides, small molecules, antibodies, even viruses. And so delivering those types of cargoes is virtually impossible. And so our goal was to kind of really be able to do that very well. So we can deliver any type of small molecule proteins because it's simple diffusion. So we don't have a charge-based system similar to electroporation, and size isn't really a limitation for us either. And so and then RNA is another kind of bread and butter for us, whether it's mRNA, siRNA, circular RNA, which is a more stable version of mRNA that could be useful, especially in the CAR world, in the cell therapy world. The one limitation that we do have is DNA. We deliver things into the cytoplasm and not into the nucleus. But that being said, you can get you can actually co-deliver a chaperone to shuttle the plasmid into the nucleus, and then things like RNPs already have an NLS on them, so it doesn't really matter. So we've done a variety of different comparison studies. I'm not going to go into everything, but here are some highlights. So this is a microarray that we did, kind of looking at global transcriptomics, and the more red and green that you have is probably not good because that means unwanted genes are either being upregulated or downregulated. And so you see that in electroporation that you don't necessarily see in mechanoporation, which is the technology or the approach that we have. And then T cell, these are in primary T cells, and so proliferation isn't affected as well. We see the same thing in HSC, CD34 positive HSCs, and we've actually done engraftment. And so our approach doesn't affect engraftment in a mouse model, where electroporation does. And on the bottom there is we preserve the stemness of iPSCs, which is a big deal because you don't want to spontaneously differentiate those iPSCs when you're working with them. So as I was saying earlier, we have three kind of different products. The first product is our gateway system, which is our R&D system. You can still do 100 million cells in less than a minute. But basically, it's the size of a tissue box, and then there's a consumable cartridge that houses that silicon membrane that I was talking about earlier, and so you just replace that after every 100 million cells. Just to give you some highlights of the data that we're able to generate, either internally or through our partners. So these are unstimulated T cells, which are typically harder to transfect than activated T cells, but this is a simple kind of mRNA delivery into unstimulated T cells, viability, and then 93% efficiency of delivery. One of the cool things that I was saying earlier is we can co-deliver multiple payloads at the same time, and three kind of completely different payloads. So here is we took T cells, we delivered a CRISPR RNP for a B2M, an mRNA expressing GFP, and a dextran, which is a sugar polymer. So three completely different types of payloads, and what we see is 86% of cells get all three. And what's cool with us, our technology is, if you look at the kind of the purple box, like you get very little single or double positive cells in this case. So if one payload can get in, that means all the payloads can get in as well. We can do mixed population of cells, so these are PBMCs. Again, mRNA delivery, and so we get delivery into Ts, Bs, NKs, and monocytes at the same time, not compromising viability. We can do sequential delivery. So one of the downsides of electroporation is the recovery time, which sometimes takes 48, 72 hours. For us, like within an hour, you can re-deliver cargo. So in this case, we took T cells, unstimulated T cells, delivered a TRAC RNP, and then activated them, and then delivered a mRNA and co-delivered an mRNA and a B2M RNP. And even you can kind of consider this as like independent events, and so we still, even in those independent events, we get over 50% delivery of all three payloads into these T cells. We can also do, so like in this particular experiment, like the delivery of IL-2 and CD19 CAR. So here we took unstimulated T cells, activated them, and then added, and we call boost, that's our lingo, but boosted them with two circular RNAs. So circular RNAs are more stable than mRNAs, and they don't have the pseudouridine modifications. And so we delivered a CD19 CAR circular RNA as well as a membrane-bound IL-2. And we did a cytotox assay for T cells, kind of looking at effector target ratios. And we see once both were administered, we see a huge bump in cytotoxic activity of T cells. Just to kind of give you a sense of the high throughput system, so this system typically is geared more towards drug discovery and screening. But it's compatible with 96, 384, 1536 well plates. And so the idea is you have two options. One is, if you look on the right, one is like we actually have an instrument that's the size of a tissue box that you can either manually utilize for kind of aspiration and putting it into weld plates, or you can kind of integrate that into a liquid handler. And so we co-launched this with Eppendorf a couple of months ago. The other option is you can actually inline integrate with dispensers in your lab, right? So whether it's a Hamilton, Tecan, Agilent. So the idea is, if you look in the purple box on the left, it's our consumable cartridge that integrates inline into a dispenser. And so the idea is you dispense kind of permeated cells, and then so you have your kind of cargo that's preloaded onto a weld plate, 96-384, and so you dispense permeable cells into each well, and so now the cargo can be uptaken by the cell, and now you can do kind of a variety of different screening. If you want to do cell-based assays or you want to grow the cells, you can do whatever you want with them. But the idea is this is now compatible for kind of screening, kind of doing a variety of different types of experimental conditions within the same experiment. And just to kind of give you an example, like we even, looking at randomly plate 2 to plate 7, you can do about 5 to 10 plates per cartridge. We still get that kind of consistent delivery, and so there's not a lot of dead volume there. So the clinical scale integration. So this is what we're really excited about. We actually have early adopters for this already, but we're actually officially launching this next month at ASGCT. But the idea is it's our millibooster cartridge. It's just a consumable cartridge that can actually integrate into an existing cell processing unit. So we've partnered with a number of companies, 2 of them being Fresenius Kabi and Thermo. So in this picture, we've integrated our millibooster cartridge that houses that silicon membrane into the Lovo to keep it a closed system. And so we're pretty much compatible as long as you have the ability to either tube weld or Luer lock a cartridge. But the idea is now, within a given cell processing unit, you now have a delivery mechanism in that same workflow that you do. And this is just some data that we have with some of our partners. So we took a leukopak here with mixed PBMCs, delivered a dextran, which again is a sugar polymer. This is representative data from the Rotea that Thermo has. And so viability is not compromised, and we're getting high efficiency of delivery of that particular molecule. We published a white paper with Scale Ready and Fresenius kind of to demonstrate our ability to deliver cargo at the clinical scale. So this is just one of the experiments that we did where we took T cells, we boosted them at the research scale with a B2M RNP. We then put those cells into a Wilson and into a G-Rex to activate them, and then using the Lovo from Fresenius and our our integrated millibooster cartridge, we at the clinical scale delivered an mRNA. So we're talking billions of cells. We see kind of a normal cell expansion, high viability. We see B2M knockout, as well as very high expression of mRNA at the clinical scale. And so just to kind of give you, like, one of the big things that we wanted to make sure of is the data that we're generating at the R&D scale the same that we're going to be at the clinical scale. And so we were able to show this at a GMP level, where each square, kind of each dot there, is a different donor. So we're getting kind of that high viability and efficiency that we see at the clinical scale that we normally see at the research scale as well. So basically, where does this all leading, right? So our goal is to kind of really think about broadening kind of the therapeutic possibilities through this idea of multiplexed engineering, right? So here are some of the concepts that we're working on with the collaborators, with our partners. Delivering membrane-bound IL-2 for increased memory function for T cells. Eliminating preconditioning. So we've done a lot of work with HSCs to deliver BCL2 and membrane-bound IL-2 to enhance survival. We can enhance killing activity of T cells by either delivering IL-2, IL-12, but also granzyme B and perforin. We can also enhance transduction. So if you're really interested in viral vectors for B cells, we can deliver a T cell receptor into a B cell to basically make them more primed for viral induction. So that's we're working on that with one of our partners. And then I didn't get into this, but we can do a lot of directed differentiation from iPSCs to a variety of different cell types. So I'll stop there. Our goal, so as I said earlier, we don't really have a limitation on cell type. Our scalability is we can do 1 billion cells or billions of cells at the clinical scale. Cargos don't really matter to us in terms of size or anything else. And so we would love to kind of broaden our ability to work with folks like you guys that would be interested in our technology. Our goal is to increase, we have 64, 65 partners right now, and so our goal is to kind of continue to kind of raise that. So I'm a little ahead of time, so happy to take any questions if anyone has them.
Thank you.
Thank you.